Streaming live from Kingston, Jamaica. It's CSEC Biology Live with Mr. Wilson. It is a cold Friday evening. A lot of persons are in home this evening. As is usual, we are going to be taking the show to the road. We are looking primarily at the digestive system this evening. Grab your friends, grab your colleagues, call somebody, and let's go with the show. My usual friends, just say hi as you log in. Now, let me just do my roll call very, very quickly. Jamaicans, are you here? Good night, Otisha. How you doing? So Guyana is in the house, checking in very early. Otaku, I'm not too sure. Otakus. I don't know how to pronounce that. Are you Jamaican? Good night. Forgive me if I can't pronounce it. Hi, Leandra. How you doing? Otaku. I'll just stop right there. At Otaku, are you Jamaican? Cindy. Okay, Cindy. Okay, I'll call you Cindy. Cindy. That sounds pretty Jamaican. Cindy. Oh, Guyana. Are you here with Otisha Gray? Or are you just by yourself? Cindy. Otisha invited you. She's always here. Otisha, Tunisia. They are always here from Guyana. Welcome to the show. It is our usual show. I bring her assist. Okay. Yes, sir. She did. Okay, so I can just use the Jamaican lingua. Big up yourself, Otisha. Yes, you brought somebody with you this evening. Tomorrow we are 3 months old, and I'm very happy that you're here for that 3 month old celebration. I am very happy. All right, so I was doing the roll call. We called Jamaican. Leandra is here representing the Jamaican. Do we have any other Jamaicans who would love to say I? Four more to go, lol. Yes, four more to go, and I hope those four will be in this evening. Leandra Burgess, who did you bring with you this evening? Did you carry anyone with you? Your friends from another school or from the school that you attend? This evening we are bringing out our friends. Is there anybody here from St. Vincent, from St. Lucia, Grenada, Trinidad and Tobago? All my families out there, my friends out there, we are waiting on you to get going. So it's another wonderful evening. We have been working. I will be telling you about the labs that I have online, the virtual labs. And we have a couple of labs that I'm always saying that we have a couple of labs because we've been working behind the scenes. We have somewhat slowed down the production of notes and that type of thing because we understand that in most cases, schools are out. You can't do labs with reagents. So we have to be doing those labs with the reagent and color change and posting them. We have been doing some nice simulation, persons have told us. So we would have had the germination lab out now. That's online. You could just up on over before we get started and look at our germination lab. It's a full simulation. I think about 5 test tube are about that. We're using pyrogallic acid and of course light and dark condition, different temperature, so on and so forth, been tested. Now we have completed, I said the last time when we met that I was working on ecology. That lab is completed, fully simulated. The quadrats are thrown, the slides are prepared. The only thing that is left to be done for that quadrat lab sampling, you don't have to go on a field trip anymore because I thought about our local syllabus and I did put in a number of stuff. You will observe that there's a river in this new settings that we have for the animation. There's a windmill. We have like the egret. We have the cattle in it. We have some grasshopper. We have some worms. We have a number of different flower. We have a trail in there. There's a road in there. So there's a whole lot of things to look at. We can look at the anthropogenic impact. We could look at the symbiotic relationship there. We could look at renewable energy in this lab. We could look at, of course, so much stuff. So much can be captured, so much can be taken from this lab. And I think it's a lab that you're going to love to see when I release it tonight. I'm going to be uploading it tonight. Yes, we did osmosis, but I've been so busy. We've done that since week and I have not actually uploaded it. The soils lab, let me show you. I'm very serious. I have your samples right here with me. The samples will be, I've been working on this over the weekend. So you see, I have been working. So this will be take the form of a clay. All right. And here we have our gravel. If you observe, the particles are really, really far apart. So we really have some sample here. And we have some sand. All right. So this is another lab that we're going to be working on. It's a real lab. And here we have loam. So this lab will be going on the road pretty soon. So we're working on a whole lot of labs. We want to ensure those persons who are going to be doing the exam, at least they can refer these labs to their teachers and their friends. And if they are even not going to be using my simulation, it is well explained that it will be helping them to complete their labs. And in some cases, persons can start to send something in the chat. If you see a lab there and you needed something, you could send something in the chat. And usually I do answer those questions. So we have been doing well. Thank you very much. We have been growing. We have been growing. I can't tell you how impressed I am. We have been growing. You have been spreading the word. You have been inviting your friends. And of course, it's because of you we are where we are now. I tell you, I am so energized. I am so fueled. I sit every day and I try to write new animation because I want students to see what is happening. Science is a little abstract for most of persons in the Caribbean. And the complaint that we get here in Jamaica is that when we use those videos from overseas, they say they talk too fast. I don't understand them. So we tend to use the local tongue, the Caribbean tongue. We tend to speak much slower than the Europeans. So we are making our videos. The quality will get better as we go along. You know that the quality will get better. But I tell you, look out for this lab that is coming out this evening. The lab that is on ecology. We are using the quadrant to sample. I have worked out an entire a situation where I looked at I think the nut grass. I sampled for the nut grass. I have done the density calculation. I've done the population size calculation. I've done the percentage frequency calculation. And of course, I will be explaining in details what is happening in this ecosystem. And of course, when you get to the point of doing your lab, by watching my lab, you should be able to, of course, write up a wonderful lab for your SBA. I'll be putting out videos for labs, because I'm pretty sure that the exam will be somewhere about June, July. And the exam being June, July, and for many persons, schools are out and you're not going back to school. Even if you were supposed to do these labs upon your return to school, it is going to be a little difficult. So what I'll do, I'll tell you about the labs I have online very quickly, and then I'm going to up on into some past papers. We're going to look at some past paper, and of course, having completed just some few questions, you too can go on my channel. There are past papers there. You need to start working these past papers because you can answer these questions because we would have covered them in the lessons that we have done so far. All right. So far we have online diffusion lab. We probably have more than one diffusion lab online. All right. We might have animation and we might have the actual lab. The osmosis lab, I told you, we have it here just for me to edit it and upload it. All right, so we have several photosynthesis lab. Let me tell you the photosynthesis lab that we have online that you can go and check. All our labs are written to satisfy the need of the C6 syllabus. Go on over there, check them, check the syllabus, and see if I'm lying. I follow it to the T. These are not just labs that we have just done these labs because they are labs. We look at what the CXC syllabus wants us to do, and we coin the lab to fit the syllabus. So we have for photosynthesis, we have the evolution of oxygen coming from the leaf during photosynthesis. That is there. We have tested the leaf to see if chlorophyll is needed for photosynthesis. That is there, and that is a real nice lab. Then we have one that I think we just test starch. That is also another wonderful lab, right? And so that's 3 labs, and we have a real lab that we did test a variegated leaf and a carinilla leaf. That's a real lab that is online as well. We have uploaded germination. Germination is on. We have uploaded a respiration, one of the respiration lab with lime water turning milky. I think you must tell me about the color change. You like the color change for that lime water thing? And then we have this lab now. We have what lab? We have respiration lab. I am working on sorting out one of the color because the color is very, very important. So I'm working on sorting out the respiration lab. everything else in the lab more. That just one color I need to sort out and things will be fine. And I also have I told you about the Ecology lab coming out tonight, and then we have another lab, photo respiration, coming out. Once I would have put out these by about Saturday evening, Sunday evening, next week I'll be looking at food tests. So I'll be doing food tests and I'll be taking you through the various color change for food tests. What I have done is to actually collect some of those lab books that we have used over the years, and I've looked at some of my better labs, and we are putting those together. So you're in good hands. I am paying keen attention to those 8 labs in the it's 8 or 7 labs in the back of the syllabus. Those will be completed. The only lab that we can't do the implementation for you, but we are going to be uploading examples of course, planning and design, and an example of the implementation lab is in the back of the syllabus. So you want to ensure that you get a copy of the C6 syllabus. It is online, it is free. Download it and look at it. If you write a good implementation lab, we are almost certain that you are a 1 student, because the implementation somewhat covers the entire syllabus. All right, so those persons who are out there load and locked in, there's not many persons here right now. We're still waiting on the others. But for those persons who are in what I like to call Yonderland, if you have not yet subscribed, please feel free to do so now. You could just click on the icon that says CSEC Biology TCP, and that should take you there. You're going to need to have a YouTube account in order to be a part of a live chat, the chat session. This is probably the only show that encourages persons to be wrong. And of course, we fix that wrong so that persons can start being right. Right? We don't allow persons to leave with misconception. We work on it as much as we know how. It's a wonderful show. It's a family show. It's a Caribbean show. This is a place where all views can contend. We're now going to be looking at a past paper. I'll be shouting some questions to you, and of course, you know how this go around here. You are going to be answering these questions, A, B, C, or D, so you'll have to listen carefully. You'll have to listen carefully. I didn't tell you that we're working also on the enzyme lab. That has been worked on, and we're looking at what is in the syllabus, and it will be sorted out. So I want you to go on over. Watch the videos, watch them to the end, give them some likes, leave some comments, let me know what I need to touch up on and what I need to, that type of thing. You follow me? Don't just allow the videos to sit there and you're not watching them, you're not sharing them with your friend. Don't be selfish. I am not selfish. I am here with you all 3 days of the week that I promise. So you don't be selfish. Share the videos with somebody else, all right? And let us grow. All right, let us look at what I have here. I think I would have had these questions online. I did answer this paper online. It is a 2014 biology May-June paper. I did answer it online. I'm going to be asking you a couple of questions now before we get into the show as we wait on other persons to join us. So I am going to go to, I want to start from photosynthesis. And it's nothing big. I'm sorry I can't show you my screen, but pretty soon we'll be able to. So it says item 8 refers to the following diagram, which represents a metabolic process carried out in plant. Now, the metabolic process in plant, I will describe the diagram for you, so you have to listen carefully. Now, the diagram has an arrow point showing that water is entering a cell, and that area that it is entering, it contains chlorophyll, and this chlorophyll is in the chloroplast. Light is also entering this area. Carbon dioxide is entering the plant cell. It is a plant cell. And of course, leaving the plant cell is oxygen and food. So there are 3 stuff that's entering the plant cell, and this plant cell contains chlorophyll, of course, in the chloroplast. Water is entering, light is entering, carbon dioxide is entering, and of course, oxygen is leaving and food is leaving. Now, the question for this, what you're supposed to answer here is the food produced is, so they want you to tell us now what food is being produced, because it comes off as food, right? So the food produced is fat, starch, protein, or amino acid. You would know what process that is if water is going in, light is going in, carbon dioxide is going in, and you know what we test for when we test this thing. So what is being produced? Is it fat? Is it starch? Is it protein, or is it amino acid? So A is for fat, B is for starch, C is for protein, and of course D, amino acid. Let me see the chat now. I guess it's on fire because everyone would have the answer now. What's your answer? Let me see in the chat. Is it A, B, C, or D? So the question is, we spoke about this diagram that has chlorophyll in it. It water entering, light entering, carbon dioxide entering, and leaving is oxygen and food. Now we want to know, what is this food? The food that is produced, is it fat that is produced? Is it starch that is produced? Is it protein that is produced, or is it amino acid? Now it's A for fat, B for starch, C for protein, and D for amino acid. What's your answer in the chat? Okay, so here we go. Yes, she did. Okay. Amoy Lawrence. Hi, Amoy. How you doing? She's Amoy saying starch. Otisha Gray is saying C, and C is protein, Otisha. I suggest that you revisit that question, Otisha. Likia Robertson. Are you Jamaican? Starch. Just tell me, Leah K, if you are Jamaican. Otisha, I am unsure. Well, Otisha, try something else. That is not the answer, Otisha. Try something else. Angel Moore is saying B. Let me see what is B. Angel Moore is saying that it is starch. All right. Good night, Moore. B, all right. Cindy is saying that it is B. Is there anybody else who would want to give it a shot? I'll read the question again, and then I will, of course, tell you what's the answer. So we're looking at this diagram. I could just draw the diagram. I don't even know where my papers are. I think I used everything is all moved up. So I have this diagram here, and I'm going to be showing you the diagram very quickly. Probably if you see the diagram, it would make more sense. All right, so it has something like that. And here we have chlorophyll. And entering this diagram, pretty much like the animations I have online, you would have CO2, you would have H2O, and of course you would have, I think it's light, right? And coming out, you would have O2 and food. It says food. Now, what we want to know is, what is the food? So I think it is something like this. If you can see, CO2 there for carbon dioxide, H2O for water, and the light there. And coming off, we have O2, which is oxygen. And what we want to know, what is food? What is being produced here? And of course, this round thing here is a cell. Many persons said that the answer was starch. And starch is the same thing as B. So let us look and see if there's anybody who would love to change what they have, or you're all comfortable with what you have. Yes, sir, I'm Jamaican. All right, big up yourself. Go and big up yourself. Yes, the answer is going to be starch. The answer is always going to be starch. I know that you are saying that, but sir, glucose, photosynthesis is glucose. But glucose is carbohydrate, a form of starch pretty much. So I say starch because plant contains starch grain. So it is in the cell, and excess is going to be stored as starch granule, and if it's animal cell, it's going to be stored as glycogen granule. But remember, the plant will produce this glucose, and it will be stored as starch for some plant, and for some plant, it will be reduced in sugar. But the most suitable answer here, remember, with multiple choice, it is always about suitable answers, right? So we know that glucose is a form of carbohydrate, starch, pretty much the same thing. And we know that glucose is the first thing is going to change is really starch. So you wouldn't want to choose amino acid. That is the simplest part of protein. And you wouldn't want to choose protein. Neither would you want to choose fat. So the most appropriate answer there, of course, would have been B for starch. Let us up on to the other question. All right. So B is the answer for that one. Let us look at the other question. This one is going to be a little hard for some persons if you did not watch my earlier video. Which of the following is not an autotroph? Now remember, autotrophs are organisms that are able to make their own food. So here we have algae, fern, moss, and fungus. Which of those organisms would not be an autotroph? Would it be A, fungus, B, moss, C, fern, D, algae? We are coming from the 2014 CSEC past paper, biology past paper, paper 1. What is your answer for that? Which would not be an autotroph? Would it be the fungi? Would it be the fern? Would it be the moss? Which would it be? Amor Lawrence is saying fungi. Is it the moss B? So fungi is A, moss B, fern C, algae D. What is the answer? Keep it flowing for me. All right, Otisha is saying fungi. We just completed that one, so we would have looked at this twice. All right, so what's the answer there? Some persons are saying fungi. Some persons are saying, I think what it is. Remember, we just looked at autotrophic nutrition, heterotrophic nutrition, and saprophytic nutrition. And also remember, last week or last class, I drew some circles on the board with some lines, and we were looking at these organisms. So what it is, which of these organisms is actually not an autotrophic organism? Any other answer? Tell us and let's go. Any other answer? All right, I'm not going to stress you out. It means that I have to go back over that bit of the lesson. So your answer is going to be fungi. Now, for those persons who have answered to say that fungi is an organism that is not autotrophic, what is a fungus? If we were supposed to look at nutrition, what's a mode of nutrition? Not autotrophic, so what it is? Let us remind the others. So Amoy is saying fungi. Leandra is saying fungi. Otisha is saying fungi. Moore is saying fungi. And my friend from Guyana, Cindy, is also saying fungi. All right? But what is a fungus? If it is not a autotrophic organism, what it is? Come on, guys. Let's move the show a little faster. What it is? All right? Let me make it very easier for you then. If it is not an autotrophic organism, is it a saprophytic organism or is it heterotrophic? Or we would just put it as heterotrophic. Of course, saprophytes are also heterotrophs. But would we classify it as just broad heterotroph or would we classify it as saprophyte? Which would we classify it as? Amoy is saying B. So does B mean saprophyte? Amoy, does B mean saprophyte? Amoy? The second one. Alexandra, how are you doing? No, the next one. So you are saying that a fungi is actually a heterotroph. That's what you're saying? I don't know if it is the service why it's moving so slow. But it's a little slow. All right, so let's treat with that. When we looked at nutrition, we said that we could look at nutrition as being autotrophic or heterotrophic. Your syllabus wants you to look at nutrition in terms of autotrophic, heterotrophic, and saprophytic. Of course, a saprophyte is also a part of the heterotrophic group. However, to answer my question, the most suitable answer would have been saprophytic. Because saprophytic organisms, they are feeding on dead and decaying organism, and they are digesting the food outside of their body. Remember, I've always been saying that. So the fungi can't make its own food. It relies on something else for food. And you'll see fungi growing on dead and decaying stuff because it's breaking it down and absorbing the nutrient, that type of thing. So we don't want to forget that. Let me look at the other thing. The fern, the fern is a type of plant, right? In classification, you would have looked at that. Algae, if you looked at my photosynthesis, my first photosynthesis lab, you'd see the euglena and the algae. They pretty much operate the same way. They have chlorophyll and they are able to carry out photosynthesis. The moss is the same thing. Like if you're in Jamaica, wherever water is running for a little time, what you'll find happening, you'll see this green thing growing. It looks like somewhat slime, but this green thing growing in the water, that's moss, you know. That's moss. And this green thing is capturing chlorophyll. It's sorry, oh my. This green thing is capturing sunlight as it has chlorophyll. All right. So we want to make sure that we are following the syllabus. We don't want to dump any knowledge because we are going to keep using them, going back for them from time to time. Let's look at another question and see how you'll fare off with this one. This one should be a little easier. This is another lab that I have online. This lab is online. Let me read the question for you. And this is a 2014 paper. And might I say, I have seen this lab on several papers. Let me tell you the lab. It says item 10 refers to the following experiment. A green and white variegated leaf is exposed to sunlight for 3 hours and then tested for starch. The result of a test are shown in the diagram below. Now, what it is showing is that before the leaf had green and white. When it was tested after the experiment, the area that was green returned a blue-black color, and the area that was white, it took the color of iodine. It took the color of iodine. Now the question is asking, based on the experiment, which of the following conditions is necessary for photosynthesis? Now, listen, it's a based on the experiment. Listen to what the question is asking. Based on this experiment, which of the following condition is necessary for photosynthesis? Look at this question, or this question wicked now. A, light. B, water. C, chlorophyll. D, carbon dioxide. The question is asking, based on this experiment, which of these would be necessary for photosynthesis? Is it light, water, chlorophyll, or carbon dioxide? Let me see what the chat is saying, because this must be a little provocative. What's your answer over there? What's your answer? Now, this is one of the lab, one of the lab that they say we should cover, one of the lab I tell you I have on the channel. Right? So repeat the experiment. Beautiful. I'll repeat the experiment. It's so easy, and it's like a off the tongue thing. They have two leaves. I will draw them for you. They have two. Which other paper is this? I don't want to mark up my stuff. All right. So I was using this. I'm going to be drawing. No, you're going to see the back of that. So remember my white paper thing? I always have white paper. So I'm going to draw the leaves. So right there, and I'm just going to draw a leaf like that very quickly. So I'm drawing the leaf to show you what it looks like. All right. So here we have the leaf before. So where we have the line, the stripe line, that's a green area of a leaf. And then this blank area here would have been the white area of the leaf. Now, after they did the experiment, what they found out is that these cross area that we had earlier, they took on, they pretty much changed to blue-black. They changed to blue-black. And the area that was white took the red-brown color of the iodine. Took the red-brown color of the iodine. So this was before and this is after. Now, based on this experiment, which of the following is necessary for photosynthesis? Is it light? Is it water? Is it carbon dioxide? Is it chlorophyll? Which of these suggestions would have been the answer? Follow the question. It's a very tricky question. You just have to know it to answer it. If I can help you, all of those are necessary for sunlight. All of, what am I doing this evening? All of those are necessary for photosynthesis. All of those. All of the suggestions there, they are necessary for photosynthesis. But what is the most suitable answer for this particular question? And that is why we keep saying that you have to know your stuff. Which is the most suitable answer? Nobody's answering. Alexandra is saying sunlight. Nobody wants to attempt? Most suitable? The plant was already exposed to sun for 3 hours. We know that. But despite the plant was exposed to sun for 3 hours, having tested it for starch, there's no starch in the white area of the leaf. What was lacking in the white area of the leaf? What was lacking in the white area of the leaf? What could have prevented the leaf from carrying out the process of photosynthesis in the white area of the leaf? Come on, answer me, guys. You know I went through this with you. Chlorophyll. Chlorophyll. Leandra is saying chlorophyll. Good. So, for this experiment, if you go to my channel and watch the lab, you will see once it's the variegated leaf, they are testing to see if chlorophyll is required for photosynthesis. This lab is straight from the syllabus. It doesn't require any thinking. From you see the lab, you know, oh, variegated leaf, chlorophyll them a test for. Let me read and see if they're trying to trick me or anything like that. Always remember that. All right? So in this particular question, all the suggestions were correct, but we had to look for the most suitable answer to ensure that we secure that 1 mark. All right? Let's go for another question. So we see we could go in the exam and we come out thinking that we did well, but we didn't pay attention to detail. Here's another question. I don't think that we did this, but we could attempt it. It said when enzymes are boiled, they are unable to function. This is because an increase in temperature. This is because an increase in temperature, A, destroy food materials, B, increase the rate of the enzyme action, C, denatures the protein of the enzyme, and D, convert complex sugar to simple sugar. So they boil this enzyme, and of course it was unable to function. Now we want to know, why was it unable to function? Was it because A, it was destroyed, the food material was destroyed? Look at this very careful. B, increases the rate of the enzyme action. C, denature the protein of the enzymes. D, convert complex sugar to simple sugar. What is your answer here? Really interesting one. Otisha is saying C. Tanisha is saying C. Tanisha, denature the enzyme. So that sounds like everybody's, a lot of persons are saying C. What is C? So I denature the protein in the enzyme or of the enzyme. That is what most persons are saying. Is there any other answer? All right, let us look at this. Be reminded that enzyme is not an organism. All right, enzyme is just protein. Now, enzymes will be denatured by arsenic, which is poison, uh, if they are not exposed to the right pH. That could also affect them. Enzymes are going to be denatured by high temperature. And we could talk about a lot of other things with enzyme, and we're going to be looking at that in this show. So, what was the question? We were looking at a number of questions. For this one, we wanted to find out, having boiled some enzyme, they weren't working. So, we wanted to find out, of course, what could have gone wrong. Most persons said that the answer was, of course, C. It, it was actually C. Tanisha is saying definitely not D. So that suggests that you want me to look at why the answer could not be the others. So I'll quickly look at that. First thing, A is an outlier. It's a distractor. So I said destroy the food material. Of course, persons who did not study is going to grab onto that, because when you boil some things, it's sort of like kill the leaf for photosynthesis and that type of thing. So person would grab onto that, but that is not the right one. B, when you boil anything, it increases the speed, the movement of particles. So persons who did not study probably would grab onto this one as well and think that the enzyme would operate the same way. No, the enzyme doesn't operate like that. Of course, you're going to affect the whole enzyme and cause it to be denatured. It could be D, convert complex sugar into simple sugar. Now, where D is coming from, let me explain D to you. When we are testing like sucrose, non-reducing sugar, and we boil and add the acid, that type of thing, this is where this is coming from. So it is saying, it is trying to figure out here whether you had SWAT for the exam or you actually studied for the exam. All right, so we have to be very careful. We want to go on over, watch those past paper, listen to the explanation, and make sure that we start removing some of the wrong answers that are already in our heads. Because I have the past papers over there. I don't know if you know, just go to my channel, go into the channel and look for the past paper. There are past papers there that I have explained. All right. So Master Magic is saying, hi, sir. It is Kuana Jones. All right. It's good to have you here. I wish you were here earlier. Please make it earlier for the other show. All right, cool. So let us go again. We're going to be looking at probably two more, and then we jump right into the show of the day. You can tell me if you want me to look at some more. I will definitely do whatever you instruct me to do, I will do today. All right. Which of the following occurs as a result of deficiency of nitrogen in plants? Now, I covered this at length when we looked at plant nutrition. Is it A, yellowing of all the leaf of a plant? Is it B, stunted chlorotic growth or stunted chlorotic plant with reduced yield? Is it C, greater susceptibility to disease and insect attack? Is it D, dark green, overly vigorous foliage leading to reduced yield? What is your answer here? Let me go on into the chat. Let us try just to read it once and you try to see how best you can solve it. What's the answer? Hi, how you doing, Magic? All right, C. Amoy saying C. Otisha is saying C. Where is Leandra? Where is Natisha? Where is Kevon? Where are the others? Come on. Tanisha, where are you? Cindy, where are you? All right, so let us up on right into that question. Let's up on right into that question, right into the answer. Let us see what is the answer there. All right, so you are saying that which of the following occurs as a result of a deficiency of nitrogen in plant? You are saying C, greater susceptibility to disease and insect attack. I'm sorry, the most suitable answer here is going to be B, stunted chlorotic growth or stunted chlorotic plant with reduced yield. Check your textbook and see that I think Ann Tyndale has a nice book there that covers a whole lot of this bit. You want to check it to ensure that you have this bit locked. All right, because I see that some persons are not so ready for the thing yet, but I am happy that I'm bringing it out early, that you can see where you are and how much you need to study. What cause the yellow color. The yellow color is caused by the absence or the reduced amount of nitrogen. So when we talk about chlorotic is usually yellowing. All right, all right. We really don't go any further than that for CXC. We don't. Sorry, Tanisha, we don't go any further than that. Sir, can you repeat, please? I will give you another question rather than repeating, because we did answer that one. So let me give you another one. Let me just up and down. 13. 13. And this is what we're going to be doing this evening. Which of the following is a function of hydrochloric acid in the stomach? I think we did some of that the other day. Which of the following is a function of hydrochloric acid in the stomach? Now, A, emulsifying fat. B, breaking down starch. C, hydrolyzing complex sugars. D, providing a medium for pepsin. What is your answer there? These might be a little out of what you should do this evening. Sir, please explain the other two. The other two. All right, so the other two, I guess that is for the question above. What question that was? Explain the other two. Okay, greater susceptibility to disease. All right, when it comes to plant nutrient, you would have been aware of when we talk about deficiency disease. Now, if a plant is not healthy, then naturally, if anything at all is not healthy, then it becomes more susceptible to disease. So I think they're asking you to draw on that bit of knowledge. However, they have something specific that they want you to look at because the syllabus would have directed us to the correct answer. So if you just knew about something not having nutrient, and in your head you would be saying, boy, whoa, this sounds like a deficiency disease, so I'm going to be using this one as deficiency disease. So that is definitely out. It's not the answer that they're looking for. It's not so much to say that because of this is going to happen. The answer for this particular question is specific from the syllabus, and we have to treat with that. For D, dark green, oval, vigorous, that is probably when it has too much nitrogen. All right, so you're putting on too much nitrogen, so it has this heavy foliage growth. Probably this is sort of nice for things like callaloo and so on, but it's not the best thing for every plant. All right, so we want to ensure that we are using complete fertilizer where they are supposed to be used and straight fertilizer where they are supposed to be used for those agriculture students. So we're looking at question 13, and I think Kevin said that it provides a medium for pepsin. I don't know what anybody else says, so let me just jump on over and look at all right, so Kevin is saying D. Daniel Williams is saying D. Amoy Lawrence says that it emulsifies fat. I suggest that you revisit your answer, Amoy Lawrence. Gibson is saying C. I suggest that you revisit your answer. Otisha is saying A or D. All right, repeat the question. The question is, we wanted to find out which of the following is a function of hydrochloric acid in the stomach. A, emulsify fat. B, break down starch. C, hydrolyzing complex sugar. And D, providing a medium for pepsin. Which would you choose as your answer? Is it A, B, C, or D? Gibson is saying A. All right. The answer is not A. The answer is, of course, D. Remember, pepsin pepsin is an enzyme. Pepsin is an enzyme. And because it's an enzyme, we have to know where we would find pepsin. We're going to find pepsin in the stomach. Pepsin and renin, they are going to be in the stomach. Now, these two enzymes, pepsin is actually breaking down protein. Renin is found in babies, and it is actually causing the milk to curdlate, to form little clumps, that type of thing. So we know that in the stomach, it has a acidic pH. Now, this stomach question is almost always there. When they're not asking you about these enzymes with the stomach. They're asking you what is the function of the acid in the stomach. But what I've come to find out, having gone through a couple of past paper, well, is that the questions are always at the same position on the paper. And if they don't give you, if they are testing about this time, they might ask this year paper about the bottle, and next year they ask about the cover. But it doesn't move from where it is on the paper. Let's up on into another question, and then pretty soon we are going into the lesson. It took us a little time here, but once you're learning, I'm fine. All right, so 14. The role of respiration is best described as we haven't done respiration as yet. Which of the following is a reason for placing green plants? All right, this is a nice one. I don't want it to answer the respiration one. That is going to be too much, and I don't want it to be all turned off. I'm going to use this one as the last question, and then we get into the lesson. It says here, which of the following is not a reason for placing green plant in an aquarium? This is a popular question, really popular question. A, providing food for the animals. B, providing oxygen for the animals. Sorry. C, providing the chlorine. Sorry, removing the chlorine from the water. And D, removing animal waste, waste products. So let me go over that again. It is saying that which of the following is not a reason for placing green plants in an aquarium? A, providing food for the animals. B, providing oxygen for the animals. C, removing chlorine from the water. And D, removing animal waste, which of course is not a reason for having a plant in an aquarium. Let me see what your answers will be. All right, so where do we start from? Should we start from Otisha Gray? Were you the first person to answer? DB Kevon is saying C. Is there any other answer? Do I need to read it again? Do I need to read it again? All right, so there's no answer there. I don't know if it is the network, but what I'll do, I'll just up on into the explanation so we can come right back into the lesson. So let us look at an aquarium. An aquarium is this thing that we have about the place, in business, place, at home, all over the place, with fish in it and plants in it and a pump in it and water in it and all that fancy dancy. And we want to know which is not a reason for placing green plants in the aquarium. So let us just break these answers down. A, providing food for the animals. Yes, we are going to have fish that are herbivores, so they might very well eat the plant, so it could be providing food. B, providing oxygen for the animals. Of course, the plant is carrying out photosynthesis, so it's going to give off some oxygen. D, removing animal waste produce. Animal waste produce. What waste is produced by animals? Carbon dioxide. So you could think about that very quickly. The carbon dioxide being given off by the animals, the plant would actually take up this carbon dioxide and use it for photosynthesis. So logically, our reason, or the most logical reason here, is going to be C, removing the chlorine from the water. That is not any reason why we put a plant in an aquarium. And if you think it's not true, oh no, I can't tell you that. That is going to be cruelty to animals. But if you put fish in chlorinated water, they're going to die. So don't do it. Please don't do it. All right, this evening we are going to up on back into the lesson that we were looking at. We started chemical digestion, and we're going to continue there. We spent a lot of time looking at, of course, mechanical digestion. And if I may, I will just recap a little bit. We looked at the dental formula. We looked at the type of teeth, we looked at their function, and there was something big that we actually ventilated. That was, of course, the structural design to its function. How does the design of a teeth, the structural design, how does it enable the teeth to carry out a particular function? and we looked at the chisel shape, we looked at the sharp shape, we looked at the cusp and all of that. And we were able to name them. We said that they were, just remind me of the name of the teeth again. Just type them in the chat for me. Type them in the chat for me. Remember we went through that? Don't let me down now. We had a grand class last week. So we said that we had the incisor. We said we had the canine. We said we had premolar. And of course, we have the molar. We went through that and we said that they are responsible for mechanical digestion, which actually starts in the mouth. Everybody was typing. I think I was too quick off the mark. We said that very good. I think you guys did well. Incisor, canine, premolar, molar, and it must always be written in that order. Don't start with the canine at the back. You might end up with some problems. Start with the incisor. They're right at the front here. Canine, premolar, and then molar. You want to ensure that you're doing that like you're following the dental formula. Always start with your incisor, canine, premolar, and then, of course, molar. And we looked at the milk dentition. We were also able to look at the structure of a teeth, and we did look at the crown and the root and the dentin and all that we looked at in our last lesson. We got to a point where we looked at tooth decay and what are some of the things that should be done to ensure that you are protecting the teeth. Now, this week, we are now moving away from mechanical digestion. Mechanical digestion. I think we came up with some fancy words last time because we usually come up with some fancy words. I think one of the fancy words we had last week was deglution, where we said that deglution, you would have seen it on some multiple choice paper. They come up with these fancy, what is this word? Deglution. Deglution is just swallowing. Swallowing. And then we spoke about the mouth, and we said that the mouth is also called the buccal cavity. The buccal cavity. All right. And after we spoke about that, I think, what's her name? Addie plus or from Addie. I think she said that, what did she say? Mastication or something like that? Mastication in the mouth, chewing, mastication. We never say any other mast or any other thing. We said mastication. All right. So you want to be careful that you don't mix up these words and go off and persons laugh at you. All right. If you can't remember these big words, just stick to what you know, chewing and swallowing and all that. Then we said that in the mouth, we come up with the first enzyme there, right? And that is pretty much where the chemical digestion starts. And in the mouth would have some mucus, some water, and of course, enzyme added to the food. And the particular enzyme that we looked at then was the salivary. Salivary what? Somebody tell me in the chat. Salivary what? Salivary what? Tell me the name of the enzyme. Salivary what? Salivary what? Don't fall asleep with me now. So we looked at the salivary amylase, right? So the salivary amylase, thank you very much. The salivary amylase is that enzyme in the mouth, and it is acting on starch. So of course, the chemical digestion begins in the mouth, and the salivary amylase is helping to break down. Break down what? What does it break down? It's breaking down starch into maltose. All right. So we have to remember these things because we are going to be going on into this thing and you need to know them. I think I brought a chart to class. It was not the best of chart, but we're going to be continuing in the lesson. Yes, starch to maltose. We're going to be continuing in the lesson. Here I have a human torsus and I also have a chart. Oh, you're seeing behind me. All right, so we got this chart. It wasn't fitting for the C6 syllabus, but we went through and we were able to put some stuff to it. And then we had a timeline. We were doing a timeline in that class to remember the various parts of the digestive system. And we went through. So what I'm going to be doing now, right? We're going to revisit that timeline and we're going to see if we can look at this torsus here and see how best. I'm not going to be using that. It has all the names on it. So I want you to look here at my torsus. All right, good. Bought this some time ago. Some time ago, probably 11 years ago, I bought this, and I bought some for the workplace as well. All right. No, no, no. Remember, in the mouth, it is starch that has been broken down, and it's been broken down to maltose. All right. So you want to remember that. We're going to be looking at the digestive system now. We're going to be looking at enzymes. So if you have a textbook, it would be nice if you get your textbook out and you can highlight this, that you did cover this, so you could just up onto something else. So we're going to be removing these. Though they are not a part of our lesson today, you could still tell me what's the name. These two things here, we're going to be removing them. What are they? You want to tell me? Anybody want to tell me? Where can I get one to buy? Oh, I bought this at Giscom in Arthur Tree. Giscom, it's in, what's the name of that plaza? It's that plaza in front of uh Pablo's. Pablo's at Arthur Tree. That's where I bought this, and I think they have some more there. Probably not much, and it wasn't so expensive. Yes, it is the lungs. Yeah, they are in some box that are a little rugged, little raggedy box, like water and so on. But I knew what was inside the box. I did not want the box. I wanted this. So I bought about 3, and I gave 2 to the school, and I took 1 for myself. All right, but I bought them. I'm like that sometimes. I buy things and give them away. All right, sometime. But my broke, I can't buy myself not even water. So these are, of course, the lungs. And then this thing here is the what? Right in the center. You see it's located right in the center of the chest, and it's somewhat curved somewhat a little bit to the left side. So it's in the center of the chest here. And this is, of course, some persons now are saying that this is the heart. Very good. This is the heart. And there's a bone that protects the heart. What is the name of that bone? Right, sir? This bone, what is it? Yes, this is the heart. All right, very good. So the heart is protected by a bone that we call the sternum. Now the ribs would protect more the lungs and this stuff. This bone, this tough bone, they call it the chest bone or something like that. Correct name there is the sternum. So it's protected by the sternum. All right, very good. All right, good. So now that we are in, I'm going to be taking out the liver a little bit. I'm going to be taking this out. And then we're going to try to see if we could follow what is happening here. I do hope that you can see. All right. Very good, very good, very good, very good. So ingestion takes place here. Remember, what we're looking at is holozoic nutrition. So the food gets into the buccal cavity, and then it's going to go to the back of the mouth. We made mention of this fancy word last week, the epiglottis. And for most persons, they learned then that you are not able to swallow and breathe at the same time. Who didn't know that? You can't breathe and swallow at the same time. If you try to do that, then you are going to, you know what, you're going to have this reflex action taking place. But let us just move away from that a bit. So we have a tube coming down here. Can you see this tube that my hand is touching? It goes right up behind here and come back up to the mouth. This tube, what is it called? What is this tube called? We have to identify the parts, this tube. We must be able to label these for the exam. More the HSB student, you need to label them. And of course, the bio students, you need to label and be able to draw as well. So anybody with anything else, what is that tube called? I just touched it just now. This tube here, what is it called? It's right inside here. What is it called? One person is saying the esophagus, someone another is saying esophagus. Otisha Gray is saying esophagus. Is there anybody else who want to weigh in on this one? What is that tube? All right. O word, O word, O word. Are you seeing that O word on screen? Anybody else want to weigh in on that tube? What is that tube called? All right, I won't provoke you. It is called the esophagus. All right, it is called the esophagus. We don't want to say that O word, but it's called the esophagus. All right, and then I like to put a lot in when I'm teaching. Sometimes, yeah, it's a lot. You have to write your little notes, note briefly, and so on when I'm teaching. I don't escape an opportunity to teach. So in the esophagus, you are going to have this contraction to squeeze the food down. Contraction, and it's squeezing the food down, squeezing the food down. All right, those circular muscles, they're squeezing the food down. What is that process called? That whole squeezing down thing, what is it called? I'm looking for a P word. Anybody else? Tanisha is saying peristalsis. Anybody else? Anybody else? Other persons are saying peristalsis as well. All right, beautiful. It is peristalsis. All right, yes, it is peristalsis. Beautiful. Beautiful. I think you enjoy this lesson more than other lessons. Now we are going in to the organism a little bit more. Now, here we have this organ here. Now, look at this organ. Can I interest you in something else? Everything I'm going to be adding a little more. So, this organ, uh, if you are choking, then we should put the hand like, let me try and see if it's something like this. You'd see that there. And then I'd hug you from behind. And then I'd put it in here, put the thumb in here. And then I would press and flick up. Press and flick up. It's an Heimlich maneuver. And that's what we do if somebody is stroking in order to get them to bring up whatever it is. All right, I make maneuver. But then we want to know what is this organ that we would be provoking? This organ. What is this organ here? No, we're not at the duodenum yet. What is this organ? What is it called? Can you see it? You want me to take it out? All right, here it is. This is the organ. What's the name of this organ? All right, so a lot of persons are saying that it's a stomach. Is it actually the stomach? Check it out for me. All right, so persons are saying that it is the stomach. A lot of persons are saying that it is the stomach. All right, so it would just come right across here above the large intestine. But then the stomach, we made mention of some sphincters last week, right? At the top of the stomach, which are the sphincter we're going to have? The top of the stomach, which of the sphincter we're going to have? The one that is close to the heart, what is this called? The sphincter. What type of muscle is the heart made from? Now think about that and see if you can remember the name of this sphincter. The sphincter here, what is it? All right, I see Adi saying that it's the cardiac sphincter. Kevon is saying that it is the pyloric sphincter. So Kevon, what is the name of the sphincter that is at the end of the stomach? Because there are two sphincters. Remember what the sphincter does? It operates pretty much like the valve would operate in the vein, right? It should close the top and close the bottom so that the stomach can churn and continue the mechanical and chemical digestion. So what is the name of this sphincter at the top versus the one at the bottom? So the sphincter at the top is called a cardiac sphincter. And the sphincter at the bottom is called a pyloric sphincter. All right. And this, of course, is the stomach. So we want to go down a little more. Oh, yes, cardiac. So we want to go down a little more. The liver is also very, very important in this whole digestive system. And then the liver has connected to it what is called, what is this green thing here? What is it called? The gall what? Remember this one here? We looked at this one last week. What is it called? Gall what? The gallbladder. Is there any other suggestion? All right, so that there is called, of course, the gallbladder. And it's very important for you to name it as the gallbladder and not just the bladder, because there's a urinary bladder as well. So you want to ensure that you name it as the gallbladder. Then we want to now get down a little further. We have this here. We looked at this in our last class, coming across at the top here. So we're not at the small intestine yet, but we're getting there. This here We say that it is both duct and ductless gland. What it is? And it is very, very important because it secretes sodium hydrogen carbonate. It's very, very important. So what it is? Somebody saying that it is a pancreas. Sorry, repeat. I was asking what is this right here? It's both duct and ductless gland, and I was saying that it is very, very important in neutralizing the acid from the stomach. And a lot of persons are saying that it is the pancreas. The pancreas. Yes, it is the pancreas. Now, right here, you would have seen where the stomach would have been connected to the system. All right, so the stomach would have been connected nicely here. Now, the first part of the small intestine, what is it called? The first part here of the small intestine, what is it called? Come on, go with me now. And we are going along with the syllabus, so we're not looking for jejunium now. All right. So Kevin is saying the duodenum. Tanisha is saying that the duodenum. Is there any other answer? Duodenum. Bertram. Lawrence is saying duodenum. Francis is saying duodenum. Atisha Gray is also saying duodenum. Wonderful, wonderful. All right, so it is indeed the duodenum. And then following the duodenum, where do we go? What's the other part of the small intestine? I see that we're having some delayed transmission with Cindy. Cindy, we understand. It happens every week. There are persons who are always logging, but that's fine. All right. So we're asking now the other part of the small intestine. What is it called? If the first part of the small intestine is the duodenum, then what is the name of the second part of the small intestine? All right, so Bertram is saying that it's the ileum. Otisha Gray is saying that it's the ileum. Is there anybody else? Ileum. Gibson ileum. Lawrence is saying jejunium. We said that we weren't going there, so we're not going to take jejunium because we're not seeing it on the syllabus. All right, but of course, there's a part that is called a jejunium, but we're looking at the ileum now and the duodenum. All right, and then what happens? Sir, what is that word? What is that? Jejunium. 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 There's a part called a jejunium, but it's not on the syllabus. So I really don't even make much. I don't really mention it much. All right. So it is now connected to the large intestine. The large intestine, right? And it connects at a special part of the large intestine. What is that part called that the small intestine is connected to the large intestine? What is that part of the large intestine called? I'm looking for a C word. Come on, what is that part of the large intestine called where the small intestine is connected? Oh, I want it to be a little more specific than colon, because this entire thing here is called a colon. The ascending colon, the transverse colon, and the descending colon. But that specific place that it is connected, what is it called? Still a C word. Come on, don't let me down now. Anybody recall? Can you recall what is it called? All right, so Adiplus is saying cecum, and some person will say cacum, and they will mispronounce it a million times, but we are talking about that word there, and of course, you have to ensure that you learn to spell it. Amoy is saying, of course, the cacum. You want to ensure that you remember that. And then on the other side, we would have the rectum and, of course, the anus. You want to ensure that you know all the parts of the digestive system. All right, that out of the way, let us just move on a little deeper Now that we understand and remember the parts of the digestive system, we are now going to be looking at chemical digestion. Particularly, we are going to be looking at the enzymes to start with. So we want to look at the three types of enzyme. There are actually three types of enzyme. Our enzymes can be really grouped into really three main group. Those that act on carbohydrate, those that act on protein, and those that act on fat. So my first question to you will be, what's the name of that group of enzyme that will be acting on carbohydrate? Then I want you to tell me what is the name of that group of enzymes that will be acting on protein. Then I want you to do the same thing for fat or for lipid. Tell me the name of that group of enzymes. You know, you can just refer to the enzyme using one group, you know. Are you writing? All right. So we have dangerous things happening here. Bertram is saying carbohydrates, and Bertram is also saying protease, and Bertram is also saying lipase. Is that true? I don't know. I'm waiting on the others. So we're trying to find out here that group of enzymes that would be acting on carbohydrate, protein, and fat. What are they called? All right, so let me see what's happening here. All right, so amylase, protease, lipase, Natisha, amylase, protease, lipase, Otisha Gray. Sir, I can't answer. I am unsure. All right, that's fine, Otisha. So the answer that we're looking for, Otisha, is amylase, protease, and of course, lipase. All right. So we are going to give you some different time. For example, when we look at amylase, yeah, when we look at amylase, we are saying that salivary amylase in the mouth is a good example of an enzyme that would fall under the amylase group. That's a group of enzyme that acts on carbohydrate. So that's what we're trying to find out. So we looked at the mouth already. So we are just going to up on into the stomach. In the stomach. So in the stomach would have, like the mouth has has saliva. The stomach would of course contain what we call gastric juice. And gastric juice is of course a mixture of hydrochloric acid, mucus, and of course renin or pepsin. Now, in babies, we are going to be finding renin, and I spoke about that earlier. Now, the stomach, I didn't tell you about the pH for the mouth. Amino acid, unsure. No, we're not at amino acid. Amino acid is the smallest form of protein, and that is how the body uses protein. It uses protein in the form of amino acid, it uses carbohydrate in the form of glucose, and it uses fat in the form of fatty acid and glycerol. So the pH for the mouth is going to be about 7 thereabout. All right. Some persons might say 6.9 to somewhere about 8, but it's usually neutral around 7. And that is where the salivary amylase operates best. And then in the stomach, we are coming from this neutral environment, and we are now in the stomach, which is a real acidic condition. What is the pH of the stomach? The pH of the stomach. Sir, 1.5, pH of the stomach, 1 to 2. Anybody else? Amoy Lawrence, 1 to 2. Anybody else? Any other answer? We're trying to find the pH of the stomach, right? And simply put, is how acidic it is at, yeah, potential hydrogen. We don't want to get there. All right, so we want to see how acidic it is. Persons are saying it's between 1 to 2. Persons are saying that's about 1.5. Persons are saying that it is, yeah. So 1 to 2 would find 1.5 between 1 and 2 and would find 1.6 between 1 and 2. Now, different books might tell you different things, but one thing we know for sure is that the stomach is acidic. It can be anywhere from 1 to 2. And the particular acid that we're having there is hydrochloric acid. However, my question to you, starch is acidic, so it's between stomach is acidic. So, all right, it's between 1 and 2. Good. All right, what is the purpose of this acid in the stomach? What's the purpose of this acid? The hydrochloric acid that we have in the stomach, what is its purpose? All right, and remember that what digestion actually means is that it's a breaking down of a food material into smaller pieces. And the whole objective is to get it to that point where the body can absorb and assimilate. All right. So with digestion, we are breaking down the bonds just the same. Tanisha, it breaks down food. All right. Kevon Max says, sir, provide a suitable environment for pepsin. Tisha, it breaks down molecules. All right. What it actually does is to one, kill bacteria. So you get some bacteria, it is sort of neutralizing them, killing them. And two, it provides the optimum pH for the enzymes to operate. Remember, if the pH is not right, the enzymes will not operate like they should. It maintain the pH of for renin and pepsin and kill bacteria. Yes, that's what it does. And of course, the stomach walls would be secreting gastric juice. The stomach walls, the walls of the stomach, the cells in the stomach wall would be secreting gastric juice. But the primary function of the acid in the stomach is going to be maintain that optimum pH that the enzyme pepsin and renin will be able to work. Remember that renin is found in babies, and what it does is to help the milk to clot. And also remember that pepsin is found in animal, and they would prefer to live in a acidic condition that is somewhere between 1 and 2. So the mouth was neutral. The stomach is acidic. What can we say about the intestine? Because digestion continues in the intestine, and then there will be absorption and assimilation. So in the Holozoic process, so far we have looked at ingestion, getting that food in the mouth. Then we got to digestion. So we have 3 more steps to go, but we have to finish digestion first. So we will have to do absorption, assimilation, and then egestion. So let's continue with digestion. So we are now in the small intestine. Particularly, we made mention of them earlier. The small intestine, about 7 and 8 are 8. Yes, it's definitely alkaline, and I spoke about that earlier. That sodium hydrogen carbonate is being secreted by the walls, not by the walls, by the pancreas. Yeah, by we call pancreatic juice. So we'll mix up stuff there coming out, and it would contain that. So of course, source. Let us look at the first part. We're going to look at the duodenum. So take off these. I wish I could share my screen. Come January, I will be able to share my screen so I could show you exactly what is happening where. All right. So just below this, we would have the stomach emptying out right somewhere up here into the duodenum. But as it comes into the duodenum there is also attached to the duodenum the pancreas and the gallbladder. Now, the gallbladder is pretty much it's from the liver. What is coming there is from the liver, right? And we'd say the gallbladder, it contains bile pigment. It contains bile pigment. And what the bile does is to emulsify fat. One would say the bile break down fat. I learned this from about grade 9, that bile emulsify fat. Bile emulsify fat. And I think I learned somewhere along the way that bile was not actually an enzyme, so to speak. All right. But it emulsifies fat. Then we get on down now. After the bile, we have the pancreas. The pancreas. And the pancreas, we have some things coming out here now in the duodenum. We have in what we call pancreatic juice that contain all of this fancy stuff I've been talking about earlier. And it contains pancreatic amylase that is acting on starch. It contains trypsin that is now acting on protein. It contains lipase that is acting on fat. But it is very important to note that the sodium hydrogen carbonate is secreted in order to neutralize the acid that is coming from the stomach. So it's neutralize the acid that is coming from the stomach so that the enzymes in the duodenum, in the small intestine, can function. And the enzymes that we're talking about are the pancreatic amylase, and they are acting on starch, breaking them down to maltose, trypsin, which is acting on protein, and of course, pancreatic lipase. And this pancreatic is just a reminder that they are coming from pretty much the pancreas. And then we get on down a little lower. We talk about intestinal juice. And now we use some big words like maltase. Maltase is going to act on maltose. And then sucrase is going to act on sucrose. And lactose and that type of thing. All right. But the bottom line here is that you need to understand that there are pretty much three primary areas that we are looking at with enzymes. We are looking at the mouth. We are looking at the stomach. We are looking at the intestine. Right? Now, when it comes to protein, I don't know if I told you that, pepsin is acting on protein, breaking down protein. Trypsin is breaking down protein. Pancreatic amylase is breaking down starch. Salivary amylase is breaking down starch. Pancreatic lipase is breaking down fat. All right? And we spoke about bile, which is also emulsifying fat. All right. So there we have a lot of enzymes. We spoke a lot about pH. I think we are supposed to be talking. I don't think we should jump on so quick. I have some other notes over here. We should be looking at, of course, the factors that could influence the activity of enzymes. I did make myself some notes so as to ensure that nothing is left behind. So I'm going to be going through those notes with you to ensure that we have that all nice and dandy before we move on into something else and leave important stuff behind. Now, enzymes are, of course, responsible for chemical digestion. Let me see what's happening here. Produce bile, break down fat. Sir, too much big words. So should we know to spell them? They are not big words, they are new words. I like to say that they are new words. And they are new words, Otisha, and as soon as you learn these words, we're going to start by learning about the stem word, the A's. So we talk about carbohydrates, the protein, right? And lipase, fats, lipid, that type of thing. And then you are going to now get into, they are very simple words. There's not a lot of word. Salivary amylase, right? Pepsin and renin. And then you go down and you're going to talk now about pancreatic amylase. And you're going to now talk about trypsin. And you're going to be talking about pancreatic lipase. So after some time, after you have practice, all will be well. When you have classes, sir? Oh my, Bertram, you are new to the room. All right, somebody answer that for me, please. All right, we're going to be looking at the enzyme a little deeper. Now, there are thousands of enzymes, a lot of enzymes, lot of enzymes, and they are really specific in the way they operate. They are not like some people who anything goes. No, they are really specific. They are business like. They are really specific. Could somebody answer Bertrand for me when I have classes? Now, the first thing we want to know about enzyme is that all enzymes are protein. All of them are protein. Once you say enzyme, it is, of course, protein. No, they are specific. We spoke about that earlier, that they are specific in what they do, and they also are required just in a little bit. We don't need a lot of enzyme. We just need a little bit to do what we want them to do. All right. So enzymes are not required in large amount. They are required in small amount. Um, I said this earlier in the lesson that they are inhibited or denatured by arsenic. We call it poison, and of course, they are denatured by high temperature and pH if they don't belong to that range. Because you don't want to say that amylase is going to be denatured by 7, and you don't want to say that uh, I've never had a class and you don't hear a yengers. I've never had a class. I've never done a recording and don't have to stop the recording to cut out a yengers passing by. All right, but that's fine. But we just went over 4 things. I want to go over those again before we move forward. All right. All right. Thank you, guys. Thank you, guys. So we have a class. We have classes those days. And if you miss the class, we usually post a live. No, it starts at 5, Otisha. It starts at 5:05, right? And we usually post the videos after, or the video posts themselves afterwards. We don't even edit it. It's real time. And then we also have a lot of other videos. You can ask somebody in the group and they'll tell you everything. But what I want you to remember, and I'm going slow on this one, help me to go very slow. We're looking at the properties of enzymes. That's what we're covering now, the properties of enzymes. The first one I want you to remember, tell me some property. The first one is that enzymes, they are all protein. Once you say enzyme, it is protein. The next thing we said about enzyme is that they are specific in nature. They're specific with what they do. All right? So you are not going to have an enzyme that is acting on protein, say coming up to act on starch. No, them not really work like that. They are really, really specific to the type of chemical reaction that they speed up. Right? And we have been talking all along about enzyme. I think we need to back up a little bit. What are enzymes? I'm sure, Otisha, somebody tell me, what's an enzyme? What is an enzyme? Oh, it is 6 in Guyana. I see. I'm so sorry. So we down in South America. All right, sorry. So you'd have to tell me what's your time in your area so I can advertise for that time. All right, so it's good thing we start at 5. So in Guyana, it's 6 o'clock. In Jamaica, it is 5:05. What's in Trinidad, St. Vincent? The other Caribbean island, you want to tell me your time? Because if our time is not fitting in, we have to just adjust that time to suit everyone. All right. So, of course, what is an enzyme? I want somebody to tell me that. Protease break down protein. Yes. Yes. Other than telling me the time, I also want to know what are enzymes. What are enzymes? We need to know that. What are enzymes? Enzyme, I want to know what's an enzyme. The activity is affected by pH. What is an enzyme? So enzymes, one would say that they are biological catalysts or they are organic catalysts, which pretty much speed up a reaction. Right? So let me see what you have there. Enzymes are biological catalysts produced by all living organisms and act on a substrate. Now they act on this, what the enzyme work on is called a substrate. Right? Like I would probably say the carpenter act on piece of board and the chef act on food. The enzymes, they tend to act on what we call substrate. All right, very good. Kevin is saying, sir, an enzyme an enzyme is a uh, biological molecule that speed up a chemical reaction. And of course, Bertram is an enzyme is biological catalyst produced by all living cells. When we talk about what is an enzyme, it's also very important in this case to add what they do. All right, the enzymes are very, very important to speed up that chemical reaction. I think I learned sometime alternate, but I have come to so accustomed to say now that it speeds up a chemical reaction. Adiplush, there are catalysts that alters the reaction time. So in most cases, I have seen these days is that they actually speed up chemical reaction. Brian, without being used up themselves. Oh yes, the enzymes are not used up. They work in the thing, but it's not like they can die. No, it's chemical, right? So we don't need them in a lot. They are able to do this work and they remain the same. A catalyst is any substance that speed up, catalyze it, a reaction, and remain unchanged. So the enzymes are unchanged. When they finish doing them, though, they are still the enzyme. These are some good notes you're putting up there. Thank you very much, guys. Thank you very much. All right. So we spoke now about one, they are specific. Two, they are all protein. Three, they are needed in very small amount. And four, we say that they are denatured by arsenic, by poison. All right. And then we are moving on now. And we could say they work best at what we call, usually you'd see a graph and then you'd see something coming around like that and a line going through the middle and right where it peaks. So really all graphs really here would have been the optimum right at the ice point just before it falls off. I'm not so sure if you saw that. So that would have been optimum pH. Now it's very important that you understand at what pH all these enzymes work, right? The popular ones are going to be like pepsin and renin, amylase, trypsin. That those that we made mention of are going to be the popular one. Now we said that they are denatured by arsenic. We said that they are denatured um, the, by pH if the pH is not right, and of course temperature, high temperature will also kill them. So we have to be very, very careful when we are working with enzyme. And when we say very careful when working with enzyme, if you don't want to get some poison, because when we talk about enzyme, it doesn't mean that in this case enzyme is like you. Because if you drink the enzyme, it's like a poison. Sorry, if you drink the poison, you shouldn't do that. Please don't. Please don't. If someone consumes that, it is going to stop the enzyme from working. So it's going to stop your life from going just the same. All right. So the body temperature is the same. If you are caught in a fire and too close, you could actually be killed. You could be a walking dead. You could just be walking and you're already dead because the enzymes are denatured. You are just like coursing out the rest of your life. And we have seen videos of these. I really don't want to go back to them because they are sad memories, and I really wouldn't want to broadcast that online. But when I have my classes, we do use some of these examples that we have seen. So the temperature, yeah. So remember all these fancy stuff about enzyme. We looked at enzyme today. We said what they were. We looked at some types of enzymes. So now I'm going to be asking you, before we move to the other part of the lesson, I'm going to be asking you just to tell me the name of the enzymes and, of course, the pH that they would work best. The name of the enzyme and, of course, name of some enzyme, where they are found, and the pH. I want you to type in the chat. And this is freelance. Just tell me the name. I want you to tell me three enzymes. I want you to tell me about protease, um, amylase, lipase, and when you do whatever is working on fat, enzyme or otherwise, tell me where it is found, what is the pH of the area, and the name of the chemical and the name of the enzyme, whichever one. All right. So I want you to do that for me. Type it in the chat now for me. All right, so I could start by saying salivary amylase, and that's found in the mouth, and the pH of the mouth is about 7. Right? That type of thing. That's what I want you to do. Or I could say trypsin is found in the small intestine and that type of thing. All right. So that would be a nice recap for me to see how much you have learned about the enzyme. Tanisha, you're not playing smart on me. Tanisha, may I have another one from you? Thank you very much. You've been so sweet. May I have another enzyme where it is found and the pH? All right. Wonderful. For those persons who are watching, if this is the show that you like, you're enjoying what is happening here, it's a teaching learning show. Everybody is learning. We're teaching each other, that type of thing. I am learning as well. And you want to be a part of the show, just click that blue button that says CSET Biology TCP, subscribe, and come on over. If you have not yet subscribed to the channel, please do so now. And if you're not able to subscribe, or you don't want to subscribe, and you enjoy what is happening this evening, you can just leave us a like or send us a comment or something like that. But we're encouraging you, if you're not a subscriber, you have no interest in YouTube, just go on over and watch our video. It is CSET Biology, the cover page on YouTube. Go on over. We have some close to, I think, 86 or thereabout videos online. One must suit your need. All right. We have videos on IT. We have VoxPox program. We have biology, human and social biology. We have several labs there. That type. We have a lot of animation there. The animations would go from primary school because we have as low as the water cycle. We have the photosynthesis animation all broken down. So even kids in primary school can access the channel. We're going to be loading almost everything bio from that level up. But it's going to take us some time, but we're going to get it all done with the help of you. All right. So Tanisha came back and she's saying pepsin, pH 1 to 2, found in the stomach. Beautiful. That's well done. And then Otisha Gray said lipase, the pancreas. What the lipase does, it's found there. pH, you didn't tell me about the pH. Amylase, pancreatic amylase, found in the pancreas, pH 78. Beautiful. Jessica, pancreatic enzyme. Don't tell me a pancreatic enzyme. That is too broad. All right. I want you to know the names of these stuff and where they are found. All right. Come on, everybody should be typing. Hi, Shakilia. We're trying now, we went through enzymes just now, and we're now recapping to see how much we can recall from what we did so that we can get into absorption. Now, we're going to be talking a little bit about absorption, and we're going to be jumping on over to malnutrition, and then a little bit on assimilation. All right, so you have to get yourself ready for us to look at absorption, and absorption is going to be the third step of the way because we did ingestion, digestion, and then we are going to be doing absorption. All right, so it's trypsin, pH about 7, found in the small intestine. All right, Tanisha, good. All right, so now what I want you to do is give me two properties of enzymes. Two properties of enzymes. Just type them in the chat for me. Let me just look over my notes and see where do I go from here. All right. All right, all we're doing there. So I want you to write two characteristics of enzymes, two properties of enzyme, whichever one you want to look at it as. Remember, if you're out there and you want to subscribe, that would be nice. We'd be very grateful. And also, if you don't want to subscribe, we have many videos there. Feel free to take part. And if there's a video that you think we should produce on biology, just send us something in the chat and we will work on it, be it animation or otherwise. But we've been doing a lot of animation because we want our students to actually see what is happening. So we are slowing down processes and showing students what is actually happening, and then we speed it up to say, well, this is the real world. All right. So we're getting there. All right, so we are getting ready for the other stage. All right, so we're going to come up on a fancy word in a moment. Another fancy word we call the villus. We'll talk about surface area. This is uh, another piece of information that we need to keep dear to our heart. After we would have looked at absorption, we are going to be looking at egestion, and of course, no, we're going to be looking at assimilation, and of course, we're going to be looking at egestion last. So let me see what's happening there. All right, so we are saying there that enzyme they tend to work best at optimum temperature and pH. Optimum temperature and pH. Beautiful. Tanisha is saying different enzymes break down different foods. So they are specific in their action. Very good. Um, More is saying they speed up the rate of chemical reaction. Good. Amoy is saying enzymes are specific catalysts, only one type of reaction they pretty much work on. Amoy Lawrence is saying the reaction of enzymes is inhibited by certain poison. Right, very good. And Tanisha is saying that, of course, the villus, they are finger-like projection, finger-like structure that absorb food, and they are actually found where, Tanisha. And then, being that you're ahead of us, I want you to tell me about the lactile as well. So you're going to be working with me on this one. I want to know about the lactile. We want to know about, of course, the villus. They are found in the small intestine, and this is pretty much a site of absorption. So when we talk about absorption, we're going to be talking about words like diffusion and active transport, because we're not going to be leaving these words anywhere. They were not taught in isolation that you should leave them when we did movement over there. As we go through the syllabus, we are going to be bringing these words again until you would have learned it all. So we are ready to go again. So my first question is going to be, what is absorption? What is absorption? It is a step in the Holozoic process, but what actually happens during absorption? So Amoy Lawrence is saying enzyme work best at a particular temperature known as the optimum temperature. And Braham is saying lactile absorb fat substance, sir. All right, very good. But let's slow it down a little bit and let us get back to the roots. We want to get back to what is absorption. That's what I want you to type on the screen for me. What is absorption? All right, what is absorption? Taking in absorption. All right. So I want you to. We don't want to leave this word. We want to get this word in. We want to get this word in. What should we understand when we say absorption? All right. So we're talking to persons who are probably in grade 7 here. What really is absorption? Sir, absorption is when nutrients move into the organism. I understand what you're saying, but to the person who understand that when something food move inside is when you swallow, I think you could be a little more specific. All right, MacLeod, uh, Gavisha. Gavisha, are you Jamaican? Absorption is digested food that passes into the blood. This is a little closer to what we're looking at. So, anything else you want to tell us about absorption? What is absorption? What is absorption? Remember, you still can call your friends from wherever they are. You still can remind your classmates that we're having the show. We have a little way to go. We have about 15 minutes leave in the real show, and then the extras come after. So after the 15 minutes, those persons but we still have some time. You can call your friends on over so we can still celebrate the content. All right, sir, my video lagging. I realize that, but never mind. We do appreciate the content as it comes. We tend to try to remember, and the members of the room, they are also assisting me. I realized that today was a real bad day. The light went, the internet went, and just about everything seems to have been a little crappy today. Amoy Lawrence, absorption is a process by which the soluble food molecules produce in digestion move into the body cell. This one looks like really nice. Kevin, you remember what we said when we said we wanted it to be specific here? Because we didn't want somebody from grade 7 to be listening to the show and they become confused to say that absorption is when you drink some water, right? We didn't want that to happen because that is actually ingestion. So we want to ensure that when we are giving these definitions, they are not definitions that can be construed to be anything else. We don't want them to be ambiguous. All right, so let me see what is happening here. Yes, I am Jamaican. All right, big up yourself. I think I'm seeing you for the first time, but welcome, 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 welcome. Those persons who are joining us for the first time, welcome. And we really want you to come back. Be reminded that in Jamaica, it is 5 minutes after 5 every Friday, Wednesday, and Sunday. If you're in Guyana, I think it's about 5 minutes after 6 in Guyana. Guyana is that only place there in Central America that speaks English. I think they might be bilingual. Otisha, do you speak any other language? I was, yeah, I think I did some reading on your country, and I think they say you do about 6 languages. Yes, I did some reading on your country. I actually have something prepared to present, but not today. All right, absorption is a process of assimilating substance into cells. Oh, nice fancy word, assimilating substance into cells. Or across the tissue through diffusion or osmosis. Here are the words coming out. All right, very good, Tanisha. Sir, it's 7:59 here. All right, it's 7:59 there. Here, it is actually 6:59 now. So you're an hour ahead of us. All right. All right, Julissa Bertram, when products of digestion are absorbed into bodily fluid. All right. Angel, are you in South Africa? Angel Moore, you are from South Africa? Oh, South America. Oh, my word. You had me there. I don't know how I saw Africa. South America. All right, never mind. Yes. Forgive me there. Angel, you are from Guyana as well, right? All right, so let us start a good show of absorption. So, absorption, we've had many definitions, and I have chosen some nice ones. It speaks to pretty much the food material having been digested. Now it is absorbed into the earth. Now it enters the bloodstream in order to go to wherever it's needed to be used for assimilation, we would call it. And if I were supposed to go back to some of these here, by which soluble food molecule are produced in digestion move into the body cell. And we had also have some other nice ones. But where does absorption take place? Help me with that. Where does absorption take place? My Guyanese are always here. My Jamaicans, they are always here. Is there anybody here from St. Lucia? Anybody here from St. Vincent? Anybody here from Trinidad and Tobago? Anybody here from um, Grenada? We have seen these persons that we want to know if they are back or they are missing the timing. Tanisha, the small intestine. So, digestion takes place in the small intestine. Now, Tanisha, I want you to do something for me, and I want the others to do it as well. I want you to tell me what is the real function of a villus and the lacteal, and what are some of the characteristics of these two that makes them suitable based on their structural design? What makes them suitable for their function? So tell me about what makes the lacteal and the villus suitable for their function. Take place in the small intestine and colon. All right. So, yes. So now I want to know what makes the villus suitable for its function. Are we ready with that? That's a big one, you know. That's a big one. That's a real, real big one. All right. So as my friends make their notes to have it posted, the villus is pretty much a part of the small intestine, and they are finger-like projections, we like to say, and they themselves has some other projection on them that we call micro, we call them microvilli. All right, and these increase the surface area of the small intestine, making the absorption process much easier. So let me see what's happening in the chat. All right. So enable substance to be absorbed more quickly. The finger-like projection, they enable substances to absorb more quickly. Lacteal is a lymphatic capillary. What does it do? What does the lacteal, what it does? Yeah, what a lacteal does? So we said that the first thing we said that the finger-like projection helped to increase the surface area of the small intestine. Another thing that we could look, if you have a diagram and you look at the diagram, you would see that it has a rich supply of blood vessels. Now you are going to need to have a rich supply of blood vessel in order to make things move very quickly. For example, if some money was supposed to be thrown away down into the town, and you had a very, very big family, your family go down into the town, they would get more money than everybody else who has just a little small family. So it is very important that we have a rich supply of blood vessel so that substances can move very quickly. Here we are saying enable substance. A lacteal is a lymphatic capillary. Tanisha, transport the food from the small intestine to where it is needed. All right. So that is the rich supply of blood vessels I think you're talking about there. Now, each villus has a network of blood capillary. We said that and we spoke about the lacteal being on the inside. Now, it's very important to note that the walls here are 1 cell thick. And you could tell me why the walls are 1 cell thick. Why are the cells 1 cell thick? Lacteal leads into the lymphatic vessel and provide a means of absorbing. Yes, it makes diffusion easier. It makes diffusion easier. It makes of course the substances move through rapidly, fast, quick. All right, so there we have a lot there being said on the villus. You are supposed to know how to draw and annotate the villus. HSB student is always about you knowing and know how to annotate it. Now, there are different parts there, and you want to ensure that you have a copy of the diagram and you are practicing. Now, I suggest to you all, once the show would have been completed, you want to watch the show again, or if I were you, what I'd do, I'd read on what was presented. Having read, then I would go back to the show, watch the show, and see how equipped I am to treat with the questions that were asked. Now, absorption in the colon. Let's talk about that. What is absorption in the colon? And you know the colon is the large intestine. Tell me about that. Absorption in the colon. Tell me about absorption in the colon. What is absorbing the colon? All right, so excess water. Colon absorb excess water. Anything else? Anything else is absorbing the colon other than water? Other than water, does the colon absorb anything else? All right, so it is excess water and, of course, mineral salts. And as, of course, the undigested food move away and go through the colon, as it move, water and salt is being removed. As such, it becomes a little harder and a little harder and a little harder. And that is why it's very important to ensure that you have rough edge in the diet and ensure that you have adequate water in the diet. Sir, what is the lymphatic about? Did we cover the lymphatic system when we looked at circulation, or we still have that untouched? All right, the lymphatic system, I think I'm going to cover it for Sunday for you. But it is that second type of circulatory system that sort of, let's say it sort of protect the body. And it is driven by bitters. When you drink bitter something, when you exercise, it helps to keep us healthy. That's as much as I would love to tell you now. Now that you have that basic, when Sunday come, I will go back to the lymphatic system and probably see if I can sort of bring it a little deeper into the lymphatic system, right? So that is what it does. It's how to help with protecting the body, and it is driven by when you drink bitters. You drink some Cersei, drink some Cinco Bible, do some exercise. It doesn't have a pump like the heart, like the regular circulatory system has a heart to pump. All right, so it is a little different. It operate a little differently. Amylase, any food that is not digested in the small intestine passes into the colon where water and mineral salts are absorbed from it. Water, fiber, and salt. All right, I'm not too sure about fiber being reabsorbed. I think the fiber, of course, will be moving through the colon because we are not ruminants, so we are not able to digest cellulose. All right, so we know now we are at assimilation. Assimilation. And what is assimilation for you? What do you understand from assimilation? Assimilation. What happens during assimilation? Assimilation. What happens during assimilation? What happens during assimilation? I'm logging. Assimilation to assimilate. What does it mean to assimilate? Tanisha. What does it mean to assimilate, Tanisha? Huh? We have many different students here. We have those who are, and we have those who are, and we have those who are. Right? So, what does it mean to assimilate, Tanisha? All right. So, when we would have looked at assimilation, we would have to go back a little further. We have to go back into the syllabus, and we will have to look at malnutrition. I'm not sure if we looked at water and energy requirements. We definitely will have to look at that. Just one thing for us left to. There's just one thing for us to look at, right? So we would have assimilation. I'm going to listen to you on assimilation, and then the next class we have that is on this topic, we are going to bring a wrap on, of course, digestion. All right? We're going to bring a wrap on digestion. I really want you to read for me on assimilation. I think it's a big topic than just a walk over. I want you to read that assimilation. I want you to look at what the liver does. All right? I really want you to look at what the liver does. But more so, I want you to read the entire topic, assimilation. Listen to my video. If you read and you come upon something that you don't understand, just post the question to one of my videos. Once it comes to me, I will do my best in answering the question and, of course, posting it so you can find it. All right. So remember, read. Those persons who are without a teacher, if I am your teacher or you are with me, you have a question to ask, just send it in one of the chat, one of the comments on one of the video. And once I would have seen it, I would have, and I am available. I will be available. I'm going to answer within a day or two. Usually I answer pretty much same time, but in a day or two, I'm like really busy. All right, so the show is not going to go tonight to 2.5 hours because I did promise you to release a video tonight. So I'm going to be taking some time to do that video so that some students I have who are waiting on it for their SBA, the simulator for the SBA, they can have it before late night so they can have it out of the way for tomorrow. All right, so I'm just going to be bringing the curtain down now. We would have stopped at absorption. So in our next show, we're going to be looking at assimilation, egestion, and then we go on over to malnutrition. Then we are going to be starting respiration. All right, we're going to be starting respiration. Now, the holidays upon us. I think most schools would give holiday come next week, Friday. I am not too sure if I will be available for the holiday period, but if I am not available for the holiday period, I'll at least make a post in my discussion. You'd have to go to my channel and go to the community section or discussion. I'm not too sure which it is for you. I think it is a community, and you will see the lineup of what is going to happen. If I'm going to have a show, I will post it there. If I'm not going to have a show, I'll let you know. But there's uncertainty around that. Why? Because I'm not too sure if I'll be in the city or I'll be out of the city. I'm not too sure where I will be. And also, I know that I'll be working on video. And of course, trying my best to see if I can get to share my computer screen with you when I rejoin you. I have not tried any new experiment with the show since we last spoke. Because when I try experiment, you know what happened. You log on to it and I have to send you 3, 5 different links for us to start the show. We don't want that. All right. So we've had a wonderful time so far. We have gone through digestion. We started with the nutrients, macronutrients, micronutrients, water soluble vitamin and fat soluble vitamin. We spoke about that. We actually spoke about mechanical digestion and chemical digestion, and we think we did an awesome job with the enzymes. Now, before I leave you tonight, I really want to do a lab with you. I want to do a lab with you. Whether you're HSB or bio student, I want to do a lab with you. We have not been doing any labs. We have not been doing any labs. I'm not too sure. So I'm going to be asking everybody in the chat now. Have you been doing labs? Yes or no? Let's just move away from the notes because the content is not, or the exam is not only about uh, answering paper 2 and paper 1. There's also the practical paper or the lab, as it were, for some students. Now, I don't want to be just preparing you for paper 1 and 2. Angel Moore, you have been doing lab. What labs have you covered so far in Guyana, Moore? Where are you on the syllabus? What are you doing? Bio or HSB? In Guyana, where are you on the syllabus now, Angel? All right, so my students, I'm going to be trying to get you. Oh, you are doing both bio and HSB. Oh, interesting. We try not to give that type of load to our kids. They're almost the same exam or pretty close, pretty similar exam. So we just allow students to do one. I don't know that they need to do both, but there are schools here that do that as well. Tanisha, labs are SBAs. You consider them to be different. Here we call it, most persons call it SB, and a teacher might say lab. What's the difference for you, Tanisha, as it were, for labs versus SBA? Amoy Lawrence, nope, private candidate. So if you're a private candidate, Amoy Lawrence, you're going to be doing the alternative paper. That is what? I know that it's paper 32 for HSB. I really don't remember what it is for bio. And if you're going to be doing that, Amoy Lawrence, it would be very important that we start helping you with things like what is an aim, how to write an aim, what is control, hypothesis, the labs that we're doing, all the labs in the syllabus, there are 42 labs in the syllabus, and we're going to try as much as possible to cover them. But more so, we're going to be covering those labs in the back of the syllabus first. As soon as we are finished with them all, I will let you know that they are all completed. All right. Gavisha, yes, we're finished. Yes, you're doing the labs. Sir, nope. All right. So, They are the same thing. Yeah, lab. Yeah, labs. We call it labs. Some person call it experiment. Some persons call it SBA. Just give me 1 second and just let me just up onto the syllabus very quickly, and let me just choose a lab from the syllabus that everybody will be doing. One that CXC wants us to look at, and of course, just run through it very quickly. Some of the expectation, as it were, for the syllabus. So I'm going to be using the bio syllabus, and I'm going to be looking at those shared labs. We did look at some lab when we looked at nutrition, we looked at food tests and all of that, and we did look at the reagent and what is supposed to be used. Now, you don't want to forget this. Please don't forget them. As I go through, don't forget them. The animations that I do for the lab, I'm trying my best now to make the animations very short. We're not going over 10 minutes with animation. Because we want you to watch them 1 time, 2 time, however many times. So we're trying to make it very, very short for you, so that when you go there, it is really beneficial. Remember that our animation, our labs, they are pretty much written for the C6 syllabus. So this is not a case where you are going just to download a lab from the internet and you're not too sure whether or not it suits your particular need. We have coined the lab to deal with your particular need, to deal with the Caribbean. Yeah, yeah. To deal with the Caribbean. So I'm going through the syllabus now, trying to get to the back of the syllabus to see if I could look at some reagent, to see if I could look at some lab for those private candidates, to see how best we could help them to master that paper, to that alternative paper uh, for the exam. When saying so, being that we have students who are doing the alternative paper, I think I need to look a little closer at the alternative paper to see how best. Usually, I give my students the alternative paper so that they can do some practice, right? They can master the lab, that type of thing. All right, so I think that was Amoy, private candidate. I suggest that you watch the labs on my channel. The animations are not in any playlist. We are going to create a new playlist for animation. So if you go on the playlist and you're trying to find labs, you won't find any labs for animation. But the animation is on the channel, right? But they are not in a playlist. So make sure that you're looking at those. Make sure that when you have some time, you just go and play a 3 minute or a 4 minute video. For those that have color change, make sure that you're following with the color change. For those that you're supposed to do some sample, we are going to be doing the transect line. I said that we do the quadrant, but we are going to try to use all those sampling stuff in animation. So these are the labs at the back of the syllabus. I am at the back of the syllabus now, and it says procedure for a candidate SBA. SBA assessment should be made in the context of normal practical coursework exercise. All right, and it is saying it is expected that the exercise would provide authentic learning experience. Assessment should only be made after candidates have been taught the skills and given enough opportunity to develop them. It also says that 18 practicals over the 2 year period would be considered the minimum number for a candidate to develop their skills, and on which the base realistic asset and which the base realistic assessment. It also says that these practical must, and it has must in bold and uppercase, include all of the following. So all of the following means that you must have an ecology lab, be it the transit line, be it the quadrant, or be it something. I would be putting up that tonight. Now we have on the channel already, it said for number 2, you must do movement, and in movement you are going to do diffusion or osmosis. I promise you, you will get the osmosis by tomorrow. The diffusion is on the channel. You can do either, but most persons do both. For photosynthesis, would have uploaded all the videos in the syllabus for photosynthesis. So you can look at that. The only one that is not there is how to draw a leaf. However, we have a drawing video that tells you what is expected for drawing. You could go there and watch that. Respiration, we uploaded one respiration video already. We are going to be uploading others. You want to look at that? The key thing for respiration here is to find out if carbon dioxide is given off. You could check on that. There's a video online for that. For food tests, we have not uploaded that as yet. For germination, we would have uploaded videos for we uploaded a video sometime this week for germination. For genetics, we uploaded the video for genetics some time ago where we are looking at continuous and discontinuous variation. Are we going to be uploading other videos for genetics? Yes, we are going to be uploading other videos for genetics, but we dealt with the most serious one first. So nobody can say that I did not get any practice, I don't know what to do for a lab. We would have uploaded that. So based on what we have on the screen in front of me now, the 7 lab that you must do, let me repeat them. Ecology, movement of molecule, which include diffusion or osmosis, photosynthesis or respiration, we did both. Food test, that is not out yet, but we're working on the video for that. Germination is out. Nutrition and disease, most persons do nutrition and disease as a PD. We don't have one out as yet. Genetics is out. So of the 7 labs, we have 5 of these labs already out. Now, I want to move on down to something else. The syllabus did say 18 labs. And of course, it says that the skill, with the exception of drawing, must be assessed at least 2 times. So with the exception of drawing skill will be assessed 1 time. So you can do more than 1 drawing, but only 1 6 you really want us to mark, right? And then must be assessed at least 2 times. The other labs are going to be assessed at least 2 times and marked. So there we have a good idea of what is required. Now, even if your teacher gave you, let me see what's happening here. Even if your teacher gave you the lab already, you would have written up the lab. Still go and watch the videos. Still go and watch the videos. And if you don't understand something, send me a message via the comment. Leave something in the comment. It's going to pop up on my screen, and I will answer at 2 o'clock in the morning or whichever time I am up. I'm usually up early. They're the same thing. I think we had that. I thought there were one type. No. There are more than one types of candidate. There are those persons who are doing it as private. There are those persons who are doing it as receipt, and there are those persons who are doing it for the first time, probably like you. Now, the private candidate, the syllabus speaks to them differently, and I do hope that my private candidate would have had a syllabus to be following and know what is the expectation. We don't want it to go into the exam and be surprised. So make sure you have your syllabus and you are following. I only see biology past papers. You don't see any lab when you go to the channel? Yeah, man, there's lab there. If you go to the playlist, go onto the channel, go to the playlist, and the playlist, I think it is the labs are in brown. But what I will do, as soon as the show is finished here, I'm going to go on over and I'm going to make a new playlist for animation where you can see the animated labs. Not only that, I will put these 7 labs from CXC in a playlist by themselves. These 7 must lab, I'll put them in a lab by them. I'll put them in a playlist by themselves. And also the other 18 labs, we are working on them. We are working on them. All right. Some of the labs are not here. We would have done transpiration already. I don't know we uploaded it as a lab, but we did it and it would have been covered in many of the videos that we did in the early part when we did classification, when we were looking at plants. And I think we had a plant with a plastic bag on it. That was transpiration, but we're going to be doing more. And for those persons who can do the lab, yes, do the lab. For those persons who can't do the lab, start off with the animation. The measurement and manipulation part, your teacher will assess. Show your teachers what we have. Whether you're a private candidate, or you're a public candidate, or you're a recent candidate, or you're a new candidate, show the content to someone. Practice, practice, practice. So I'm going to bring the curtain down this evening. I wanted to bring the curtain down with one of my quotes. I tend to write some quotes from time to time. Life send me some messages, and when I get these messages, I tend to find some nice way of course, writing them down that I can communicate it to somebody else in the future. So I am going to be reading you one of my quote. All right, so just give me 1 second there. I thought about it. I wanted to share with you my quotes before I leave. Do you have any question you want to share? You want to ask? I don't see any HSB past paper. Yeah, that is true. There's no HSB past paper on it. I didn't get any HSB past paper, but what I know, having looked at the HSB past paper and looked at the bio past paper, I have seen past paper around the place. I realize that some of the questions that are from biology, they are repeated for HSB. So it is safe to say that you can practice using the bio paper for the HSB. Of course, when it gets to the back of the syllabus with disease and water cycle and so on, it might be a little different. All right, so I'm going to be leaving you with one of my quotes, the one I wanted to leave with you. I can't find it, but I have so many. I will just leave you one. The point I'm going to leave with him, quote 59, it says, at any point, everything can start over. So if you made an error in life, if you made a mistake, if things are just not going well for you, be reminded, at any time, anything can start over. The other quote I'd love to share with you from my writing, quote 63, it says, if you keep going, you will reach. Just continue the journey. Don't give up. Once you keep going, you will reach. It might be hard, it might be far, the words might be new and big, and you're tumbling over these words, you're tumbling over the books, you're tumbling over the past paper, you're tumbling over COVID. But you can't give up, because as my quote would have said, if you keep going, you will reach. And for those persons who would have blundered in the past and you are going at it again, remember, nothing is so bad about that. At any point in time, you can start over, right? And at some point, everything will start over. So you got an opportunity to start over. Make the best use of it now. All right. From wherever you are in the Caribbean, from there are only 3 past papers. You will. Oh yes, I have 20 years worth of past paper. Yeah, we're going to be putting up more past papers for December. Amo, is your exam for January? Amo, your exam is for January? Yeah, we're going to be putting up more past papers. But one of the things with putting up videos online, and you need to understand this. If you're not watching the video and the videos are not being consumed, when we look at the back end, we realize you're not watching the video, we don't put them up. We'll put up videos that persons are consuming. Like if you're running a shop and somebody buys some flour and nobody not buying the flour, you don't go buy flour again and just make the flour keep spoiling. So the more you watch the video, if we see the videos are being consumed, we make videos along that line. If we see that persons prefer to watch the live, we of course spend more time on the live. If persons prefer the animation, we spend more time on the animation. So it is always going to be based on your consumption. So the more you watch the video that you like, it's the more we produce a video around that particular topic or using that particular technique. All right. So it's all based on you. It is supply and demand, even though you are not spending. So watch the videos, get your friends to watch the videos, because the more persons watch the video, the more the videos will be recommended and the more we will be fueled to make more of that particular video. All right. Remember, making a video sometimes takes a lot of research. I might look like a lot is in my head, but sometimes I forget a lot of things. And of course, sometimes I want new knowledge. I want to learn from other persons. So I might just look at other person's work and what does that mean here? What that type of thing. So it takes a lot of research. It takes a lot of time. More so the animation, you have to think it out. You have to get everything correct, spot on. If you don't get everything correct in a spot in an animation, you are going to have serious problem. Because, for example, if you are doing a color change and it's supposed to be blue, black, and I send you green, can you imagine what I'll be doing to you? All right, supposed to go through 3, 4 different color change, and we did not pick that up in the animation. So it takes a lot of time. So for me, from me to you, I suggest that you watch the video, you recommend the channel to other persons, you get your classmate, get your friends to watch the video, that type of thing, and you guys are family. I'm sure if you message me, once you message me, I can tell you that this person is from the show, or this person is family. I need to treat with this right away. And if I don't know who it is, I call my assistant and I say, hey, do you know this person? That type of thing. And of course, we act on it very quickly. We treat family different, Once you another show, you're family, and once you communicate, we respond. All right. So from me to you in Kingston, Jamaica, it's Mr. Wilson, CSET Biology, HSB Live, another Friday evening. This evening we covered digestion. It was indeed our pleasure to be here with you. We are so grateful that you took time out to come over here and study. Whereas so we know that you had other things you could have done. We know you could have been watching television. We know you could have been eating. You know you could have been sleeping. There's a cold front out. We know you had so many options, but you chose us, and for that we are grateful. We are still not at 1,000 subscribers, but we are pretty close. And we're sure that we're going to be getting there very soon, because all the family members, you are going to be getting. Remember your promise you're going to get me 5 subscribers last week. So I am not going to add anything. I'm not going to be adding to that number, right? So you said that you're going to get me 5 subscribers last week. So I'm going to be looking out for those 5 subscribers, right? But we are looking good on that side where subscribers are concerned. We are growing and growing and growing and growing and growing and looking very, very good. I'm very happy. I'm very grateful. I want you to have a blessed weekend. I want you to stay away from trouble. It's a Christmas period. Look around. Keep the doors, keep the windows closed. Wind up the car window. Shut the door. Don't trust anyone. If it's even somebody turn up to your house to say, I am Mr. Wilson, or Mr. Wilson send me, don't trust them. Mr. Wilson not sending anybody go to anybody else. No. Mr. Wilson not asking anybody to go and collect any money for him. No. If I never say it here on the show, it was not said. All right? So don't let anybody fool you about anything at all. I never sent anybody to collect any money from me. No. All right? So you are going to stay safe. We are going to post whether or not we're going to be having show next week. We're going to continue to send out the videos. Remember to watch the videos. Remember to share. Remember when you watch them, leave a comment number on the video you never ready, the video you did ready. Yes, the video you pop, that type of thing. Leave a little like or something, man. Oh, not treat me so bad. Me not like how not treat me. Not treat me so good. Anyhow, roll call. Now that me telling some glad some of the day on the show, whatever, whatever. Roll call. Jamaicans in the house, just say hi and bye for me. Guyanese in the house, say hi and bye for me. Trinidadian in the house, say hi and bye for me. That's how we're clocking out this evening. Persons from, I can't even call the persons from St. Vincent. Say hi and bye for me. Those persons from St. Lucia, say hi and bye for me. Persons from Grenada, say hi and bye for me. Trinidad and Tobago, hi and bye for me. And if you are new to the show, please identify yourself so we can big you up. All right, so the Jamaicans are packing in. Amoy Lawrence is saying no, up. All right. Leandra Burgess is saying up top. Tanisha out of Guyana is saying hi and bye. Amoy Lawrence from Spanish Town is saying hi and bye. Anybody else in the house? Otisha, are you still here? Are you still here? All right. So as we make our way out of the room, we catch you on Sunday, where we are going to continue with the exploits of genetics, and we are going to definitely look at that lymphatic system for Otisha and genetics. We're not going to be doing the pedigree chart at that time. We're going to do the pedigree chart about January. And when we take the pedigree chart to you, make sure that you're going to be taking it to somebody else. Until Sunday, take care.